This is part 3 and hopefully the last part of the recorded lectures for chapter 4. A lot of what I'm going over with you now, we went over when I went over the when I covered the animal and the plant cell for the eukaryotic cells. So just remember that the nucleus is the command or the control center of the cell. It does contain the DNA, and the DNA is either in a form called chromatin. Chromatin is kind of very spread out DNA. It's not condensed into any shape. It's just spread out. A bunch of very, very small strands just spread out, throughout the nucleus. But when the cell gets ready to divide, the DNA condenses, the chromatin condenses to form chromosomes. You may have seen pictures of chromosomes before. When they are duplicated, they look like X's because it's like two straight linear structures that are connected at one spot, so that it makes them look like, a bunch of X's. Humans have 46 total chromosomes in their cells. 23 came from their mom and 23 came from their dad. But anyway, chromatin and chromosomes are just different forms that DNA take inside of the nucleus of eukaryotic cells. And the nucleolus is the part inside the nucleus that is composed of ribosomal RNA. What you need to know about the nucleolus is that it makes the ribosomes, and then ribosomes make proteins. Ribosomes are the site of protein synthesis in the cell. And down at the bottom, just because there will come a time when we come back to this, when we get to the chapters on genetics and how DNA is replicated and how DNA codes for our traits, the central dogma of molecular biology is literally this little sequence right here. It's DNA produces messenger RNA produces protein. So DNA codes for messenger RNA, then messenger RNA codes for a specific protein that's going to do a certain job. So that is the central dogma of molecular biology. And again, we'll come back to it in a later chapter. I think it's chapter 10 or chapter 11. This just shows you how ribosomes work, and it's a little early, I believe, for us to be talking about how proteins are made, so I'm going to skip that. The endomembrane system, you may have heard of the cell being compared to a factory. So each cell is a little factory, a mini factory. The endomembrane system is that part of the cell that works like a small factory. It consists of the nuclear envelope, not inside the nucleus, but the actual nuclear envelope, the endoplasmic reticulum, which is attached to the nuclear envelope, and the Golgi apparatus, and then transport vesicles. And so the endoplasmic reticulum is a component of that endomembrane system. It's a system of membrane channels and saccules. Saccule means flattened vesicles, little sacs. Continuous with the outer membrane of the nuclear envelope. That means that the ER is attached to the nucleus at the nuclear envelope. The rough ER is studded with ribosomes and has the capacity to produce proteins. The smooth ER does not have ribosomes, and it's involved in the synthesis of lipids. And here's something that I highlighted again because I saw it on the quiz questions. In the testes of male animals, testosterone is produced by smooth ER. So the testes would have a larger amount of smooth ER than maybe some other areas of the body. Then we have that the smooth ER is also the site of various synthetic processes, detoxification, and storage. So again, we see this twice in this one slideshow. We've seen this twice, so it's probably going to be on your midterm. The liver with abundant smooth ER detoxifies drugs. So two places that we know have a lot of smooth ER, and that is the testes and the liver. And then this just shows you a picture. So this would be the endomembrane system part. This would be the nuclear envelope here. And then outside of that, you would have the rough ER and then the smooth ER. And then if you look at the cell, an animal cell, it goes the nuclear envelope, the rough ER, the smooth ER, and then the Golgi, and then the vesicles. That's the endomembrane system. The Golgi apparatus takes the products of the endoplasmic reticulum and packages them up. It can modify them a little bit. So it'll modify proteins and lipids with signal sequences, and then after modification, it will prepare them for shipment, meaning that it will put them into a vesicle. And they will leave the Golgi and either stay inside the cell or they will leave the cell, depending on what their job is, you know. So again, this is just another view of the endomembrane system and the Golgi apparatus, the role that the Golgi plays. So here's the nuclear envelope. Then you have the rough ER that makes proteins, the smooth ER that produces lipids, and both of these can pinch off on the ends and form vesicles, and their products will fuse with the Golgi. And the Golgi will modify those products and package them again to either be sent out of the cell or to stay inside the cell and do some, you know, play some role in this inside of the cell. Okay? Lysosomes are actually vesicles, but they're special vesicles. They're vesicles that are not found in plants. They contain powerful hydrolytic digestive enzymes, and they're highly acidic. In white blood cells, lysosomes are used to, well, okay, I see what they mean. Okay, so I was thinking the white blood cell itself engulfs pathogens, and then the lysosomes can digest those pathogens. But sometimes the pathogen, like a bacterium, will fuse with the lysosome, and then so the lysosome can engulf the pathogen in certain cases. But anyway, the lysosome basically has the enzymes inside of it that will break down and digest anything that is old or worn out or has something wrong with it. You know, it's faulty in some way. Or it's a pathogen, which means it's something that can cause disease. Now, if there are defective lysosomes, sometimes diseases can come from that. So some of these diseases are called lysosomal storage diseases. One of the worst ones, it may be the worst one, is called Tay-Sachs because it's a genetic disorder that affects babies. And but anyway, it's something that we talk about later on in the course, so I'm just going to leave it. But if you want to Google it and, you know, look it up, you can. Anyway, lysosomes are for digestion. This is a summary of the endomembrane system. I'm just going to read this part here. I think hopefully you're starting to see a pattern where it says example. You know, I will highlight these examples because I've seen them on the quizzes in Connect. So this example says mammary glands produce milk and the pancreas produces digestive enzymes. So mammary glands, the cells that form mammary glands, the product of those cells is milk. And that product needs to leave the body because it's for babies, you know, it's for the offspring. So in this case, the cells of the mammary glands are little factories that produce milk. And the cells of the pancreas, you probably think about the pancreas as producing insulin and helping to keep your blood sugar low. But also the pancreas produces digestive enzymes that helps us to digest the food we eat. Those digestive enzymes are secreted into our small intestine, and that is where our food is, most of our food is digested, is in the small intestine. Here's a picture of the endomembrane system, and I encourage you to read this slide. I'm not going to go through it because I already have, but it's a better picture than the ones I've been showing you. I will go through it just slightly. All right, so here's the nuclear envelope, here's the rough ER, here's the smooth ER, here's the Golgi. But after the Golgi, when the Golgi packages the products into a vesicle, in this case, the vesicle actually becomes a lysosome because the products inside of the lysosome are digestive enzymes. And you can see here that the lysosome can fuse with another vesicle that maybe has food in it, and it can digest that food. Maybe this is some of the food that you ate, and now the lysosome fuses with it and breaks down the molecules into smaller molecules. And another thing that can happen is the Golgi can form transport vesicles, and those vesicles can fuse with the plasma membrane and push the products out of the cell. And this is called exocytosis, which we'll actually talk about in chapter 5. But the reason the vesicles that you see them like in the pictures fusing with the plasma membrane is because the vesicle is also composed of a phospholipid bilayer, which is an oily type, a lipid type of substance. You know, it's an oily type substance. And so they can just easily fuse together, vesicles and plasma membrane. And that happens all the time. Peroxisomes are similar to lysosomes, but lysosomes are more for digesting anything, whereas peroxisomes are going to be digesting fats or lipids, fatty acids. And they do produce hydrogen peroxide as a byproduct. And so they contain enzymes to get rid of that hydrogen peroxide, to break it down. Vacuoles are like vesicles, but they're larger. You need to remember that plant cells typically have a central vacuole that stores water. There's a picture of it. Chloroplasts are found in plant cells. They have little green discs called thylakoids, and this is what the part of the chloroplast that absorbs sunlight or that captures sunlight. The thylakoids are in stacks, and those stacks are called grana. So the chloroplast has thylakoids, and then it also has stroma, which is the fluid around the thylakoids. The thylakoids are where the chlorophyll, the green pigment chlorophyll, is located. And that's why the chloroplasts are green, is due to the green pigment chlorophyll. Here you can see a chloroplast, and what chloroplasts do is carry out photosynthesis. And then when they produce the food in the form of carbohydrate, the carbohydrate goes to the mitochondria, which all cells have. Only plant cells have chloroplasts, but all cells, including plant cells, have mitochondria. That's important for you to know. All eukaryotic cells have mitochondria. Only plant cells have chloroplast. But the mitochondria will break down the carbohydrate that was produced in the chloroplast, and it will form ATP. Okay? So, chloroplasts carry out photosynthesis, and here is the chemical reaction of photosynthesis in words. Solar energy or sunlight, plus carbon dioxide from the atmosphere, plus water, usually absorbed into the roots of the plants. All of that reacts, produces carbohydrate or glucose. We usually say it's glucose, but it can be any carbohydrate. But it produces glucose plus oxygen gas, and that is where we get our oxygen on the earth, is produced by photosynthetic organisms like plants. And the mitochondria, all eukaryotic cells have mitochondria, and mitochondria carry out cellular respiration. They take the oxygen that's given off in photosynthesis and react the oxygen with the glucose that we get from the food we eat, and that produces ATP, and ATP is what our cells use for energy. So the mitochondria and the chloroplast are kind of energy-related organelles. And then the last thing I want to go over with you is the cytoskeleton. The cytoskeleton maintains, and it's not made of bones, it's made of like protein fibers, but it maintains the cell shape. It assists in movement of the cell and organelles, makes internal transport possible, and it has three basic fibers: actin filaments, intermediate filaments, and microtubules. So actin filaments, you may have heard of actin and